Hi, good evening. Due to some issues in screen sharing, the previous recorded lecture wasn't really visible, especially the portions wherein I had to write and explain it to you. So I will be taking that portion again with additional details as well about peptide bond. So let's start. I will share my screen. I hope it's visible. Just start. So uh, because we are reading about proteins. So we will be first looking at how proteins are formed, what are its monomeric forms, uh, what happens when, how are those monomeric units joined together, and how multiple such monomeric units uh, fold into fold and form specific structures, and how those structures determine the function. Now let's move on. So the general structure of amino acids, we will look at the general structure of amino acids, which is basically we have C atom here, there's a carboxyl group here, there is an amino group here, and C is here, and here this is R group, which can be any functional group. Okay, functional group. In any kind of functional group can be here. So the simplest of all amino acid, simplest amino acid is glycine, where this R group is replaced by H. Okay, so it is basically CH2, COOH, and NH2. So this is the general structure of an amino acid, wherein these two, so it any amino acid has at least two ionizable groups. Okay, so we will later come across how are these ionizable groups really significant or important in determining the characteristic or in determining the characteristic feature and characteristic behavior of amino acids in a solution or in a medium. Okay. Now, since these amino acids, we will also again look at it in some detail later. Usually, except glycine, now here this is the general formula of an amino acid. This is an R group. Now, except glycine, where both these groups are H, other than that, this particular C is asymmetric, or it shows it is a chiral carbon. Chiral carbon wherein it can show stereochemistry, like it can either rotate, it can either have L isomer or D isomer. L means levorotatory, rotatory, D means dextrorotatory. So these name dextrorotatory. Now these terminologies come from whether these particular molecules are able to convert a plane polar like a polarized light in whether it is able to rotate a polarized light into in the anti-clockwise or clockwise direction. Okay, so accordingly these are another representation of like wedge and dash representation of these amino acids, wherein two moieties are coming out of the plane of the paper and two moieties are going into the plane of the cup. Okay, and paper or the plane wherein we are representing the molecule. Also, suppose if we are keeping this CH3 group fixed here, then if the H atom is on the right hand side and this NH3 or NH2 group is on the left hand side, it is L-alanine, whereas if it is the other case scenario, it is D-alanine. Now, usually all the naturally occurring amino acids are L forms or L stereoisomer. Okay. This is found in nature in general. So, yes, like, so, yeah. So, amino acids in proteins are L stereoisomers. All right. Now, after we got to look at the general structure of amino acid, let's focus on. So, now, let's focus on what are the different kinds of naturally occurring amino acids most commonly found. So, there are 20, 20, sorry for the, yeah. So, there are 20 naturally occurring amino acids commonly found in our system. So, let's now the amino acids are actually classified based on the R group or functional group present in its site. In case of glycine, we saw that it is the simplest form with H present over here. Now, in case of other ones like alanine, proline, valine, methionine, isoleucine, leucine, all these have aliphatic or side chains which are non-polar in nature. Now, in case of proline, what we see here is it is also aliphatic in nature, but here we see the NH2 group that is there. That is actually is forming a five-membered ring here. This. Five-membered ring here, thereby giving a ring-like structure, like an unusual structure to proline. But overall, it's non-polar because here there isn't any extra nitrogen or electron donating or electron accepting moiety present as such. And in case of valine, we have two methyl groups here in leucine and isoleucine. And in case of methionine, we have a sulfur group present over here. So these are all non-polar in nature. And also one thing to note here is, so we saw that except glycine, all the other amino acids have chiral carbon. Also, because this is, see, this is the general structure. Now, this is the acidic group. So this carbon is denoted as alpha carbon. Now, these are all here, these are all alpha carbons. Okay. Alpha, alpha, alpha. And now, based on that, this will be beta, this will be gamma. Accordingly, this will be named. Now, in case of isoleucine, and also this C alpha carbon is chiral in all the cases or asymmetrical in all the cases except glycine. But in case of isoleucine, there's one more chiral carbon, which is this is the beta one. This is alpha, this is beta. So in case of isoleucine, the beta carbon, C beta, is also asymmetric. Why? Because see, this is one group attached to this beta carbon. This is one group, H atom attached to this beta carbon. This is one group attached, and C2H5 is another group that is attached to this beta carbon. So yes, like this has two chiral centers or two asymmetric carbons. Okay, next we have aromatic R groups. The R groups can also be containing benzene rings or imidazole rings, or uh, you know, like especially having these aromatic rings wherein the electron is delocalized over the plane, okay, over this region. So this can be phenyl. There are three of them: phenylalanine, tyrosine, and tryptophan. So when we see the when we talk about the absorption of protein molecule, then these are mainly responsible. These aromatic amino acids are mainly, or aromatic amino acid moieties are mainly responsible due to the delocalized electron cloud to really give the absorption spectra of a particular protein. Next, we have polar but uncharged R groups. So here we have serine threonine, which is again due to the presence of OH group here. This renders it uh, like as in renders it a polar characteristic, whereas cysteine here has sulfur attached to it. Asparagine has amide group attached to it. Glutamine also has amide group attached to it. Now again also in case of threonine here, so all these alpha carbons here are all asymmetric. Okay. But in case of threonine again, in case of threonine also, the beta carbon is asymmetric. Okay. Beta carbon is asymmetric. So threonine and isoleucine, they have two asymmetric carbon atoms. Okay. Next, we have positively charged R groups, wherein like there's lysine. Again, the naming will be beta, gamma, delta, epsilon, delta, epsilon. Like just denoting these carbon chains. There's arginine, there's histidine. Histidine has a imidazole ring attached to it. Okay. Next, we have negatively charged amine, like R functional groups, wherein it's sorry. So wherein aspartate has an extra carboxylic group here, glutamate has extra carboxylic group here. Here now one more thing to notice is, so aspartate, asparagine, glutamate, glutamine seems as if this carboxyl group has been modified to attach one amide group or CONH2 group here. But this is not the case. This isn't any modified form or an intermediate. This is naturally already present in nature or in natural systems. Okay. So these are like individually already present. It's not this is not a precursor for asparagine, or this is not a precursor for glutamine. Now, since proteins can be, range from can be composed of a monomeric, can be composed of a huge range of maybe uh, number of amino acids, which can range from hundreds to thousands. Now, for such a long amino acid sequence, we can't really put all these whole names together. So that's why some abbreviations and one letter codes were formed so as to denote the sequence of an amino acid, like sequence of a protein, amino acid sequence of a protein. So how did they really come up with this naming or nomenclature? So especially in case of the one letter code, it was like based on the abundance out of these naturally occurring 20 amino acids. Based on its abundance, uh, it was named. It was assigned one letter code based on its abundance. Suppose leucine is very abundant, so it was assigned like one letter, like a code of L. Okay. Then it was again after naming it. Now when there are like amino acids with starting with the same letter, what they did was suppose when it sounds phonetically similar, suppose in case of phenylalanine here, phenylalanine, it seems as if it starts with the letter F. So for in case of phenylalanine, it is denoted with letter F. Similarly, and then after this phonetic system, Then came just randomly picking up, um, any letter from in between the name of the amino acid, and then that was assigned as its code. So let's just briefly look at what are the, you know, abbreviations as well as one letter code of all these amino acids. Glycine goes with the abbreviation GLY. One letter code is G. Alanine goes with ALA. One letter code is A. Proline goes with PRO. One letter code is P. Valine goes with Val. One letter code is V. Leucine goes with Leu. One letter code is L. Isoleucine goes with ILE. One letter code is I. Methionine goes with Met. One letter code is M. Serine is Ser. One letter code is S. Threonine is THR. One letter code is T. Cysteine goes with Cis. One letter code C. Asparagine is ASN and one letter code is N. Okay. These are just like few, this is something to be remembered. And then glutamine goes with GLN and one letter code is, sorry, Q. What else? Phenylalanine is PHE, one letter code is F. Tyrosine is Y, one letter code is Y. Tryptophan is TRP and W is its one letter code. Lysine is LYS, one letter code is K. Arginine is ARG, one letter code is R. Histidine is HIS or one letter code is H. Aspartate is ASP, one letter code is T. Sorry. Glutamate is GLU, one letter code is E. Okay. Now, moving on. Oh, sorry. Okay. Here we see that, yeah, what I was telling earlier, like these residues are the amide form, like what we are talking about, asparagine and glutamate. So these residues are the amide forms of aspartate and glutamate, definitely. But these are, but the corresponding amino acids occur naturally and are incorporated directly into proteins. So asparagine and glutamine residues do not occur as a result of amidation of aspartate or glutamate residues in proteins, as we saw here. Okay, right. So this can be replaced with CONH2 to form asparagine, and this can be replaced, this carboxyl group can be replaced with CONH as in this, as an OH group can be replaced with NH2 to form glutamine. But this is not the case. This is already present naturally and is naturally incorporated into proteins in living beings, in natural systems. And yeah, like isoleucine and threonine have asymmetry beta carbon that we saw here. Okay, this and this. So this is or which sounds phonetically similar, then that particular letter was assigned to the amino acid. One letter code was assigned. Now, since we saw that the aromatic amino acids, due to their delocalized electron density, can really absorb UV light at a particular wavelength, wherein the highest absorbance is shown by tryptophan residue at 280 nanometer. wavelength, and tyrosine also shows some absorption. Phenylalanine also shows in this range, but then it's very less as compared to tryptophan and tyrosine. Tryptophan is the major component giving absorption. So when we are talking about absorption, actually, so at 280 nanometer, is there? So when we want to really measure the concentration of a protein after purifying a protein, or suppose when we purify a nucleic acid, whether RNA or DNA or plasmid form of DNA, plasmids when we purify, we need to check the purity of the sample for the presence of different proteins or amino as in proteins or polysaccharides or any organic compound like that, like or salts or something. So for that, we have absorbance ratios. After taking the concentration, we also check the concentration definitely, but we also check the absorbance ratios at different wavelengths. So one is like at 260 nanometer, nucleic acids absorb, and at 280 nanometer, which is what we see here, 280 nanometer. This is where the proteins show absorbance, which is primarily due to the aromatic amino acid residues. So this ratio gives us an estimate of the protein contamination in nucleic acid. Okay. Protein contamination in nucleic acid sample. Okay. Now, so what will happen is if there is more protein present, this overall ratio will be lesser, definitely. So just because this I've mentioned this, just for some trivia, it's like for DNA, this ratio is considered to be around 1.8 to 2 is fine. For RNA, also this ratio from 2 is considered very pure or good enough sample. So anyways, like at 280 nanometer, just wanted to say because at 280 nanometer, these like proteins absorption, we measure the absorption of proteins basically due to the presence of all these aromatic amino acid or amino acid residues with aromatic side chains. Now, when we are talking about absorption, we are talking about absorbance and absorption of light, then we have the Beer-Lambert law. So this particular law is based on certain assumptions wherein they assume that incident light should be parallel, okay, and it should be monochromatic. Monochromatic means only light of single wavelength can pass through, and this is an assumption that the solvent and the solute molecules, the medium in which our sample is resuspended or dissolved or something, that are randomly oriented. They don't have any specific area, or it's not specifically located in this solution. It's like randomly distributed overall. So what happens here is now if the incident light's intensity is I naught initial intensity, and after, because of the presence of some sample, after this light passes through a cuvette of this path length, L is the path length here, which is usually in centimeters. Okay, so it's usually in centimeters. So this, yeah, and also having the sample is having, suppose a molarity of C moles per liter. Okay, and due to the presence of the sample, it will absorb some amount of light and then transmit the rest of it, and this transmitted light is detected by this detector over here. Now, there's a term called transmittance. Transmittance is the ratio of the intensity of the light transmitted divided by the initial intensity. Okay. Now, log of this value is term absorbance, which has been found to be proportional to the concentration of the sample. Definitely, if the concentration is higher, it will be the absorbance detected here in the detector will be greater, definitely. And it also depends on the path length, which is fixed in this case. Okay. So absorbance is also denoted by A. So we can write the equation as A is equal to ECL. This epsilon is molar absorption coefficient, which will be having a unit of molarity liter inverse, or we can have um, epsilon, yes, sorry, not liter, very sorry, centimeter inverse. So this will be actually liter number of moles inverse centimeter inverse. This will be the sorry uh, unit of this molar absorption coefficient. So here in this case, what happens is this is a characteristic property of the sample or molecule or whatever the absorbance of the sample we are trying to measure. This we don't know. We have to actually calculate the concentration, but this is fixed and this value is given by the detector over here. So from all these values, we can actually measure the concentration of the sample, which is unknown to us. And this is how we measure the concentration of different samples or samples of containing biomolecules like proteins, nucleic acids, etc. Yes. Now, let's look at the acidic and basic properties. So the general formula for, as in general formula for amino acids is this. Okay. Now, so there are two ionizable groups. Sorry. One is carboxyl group, one is amine group. NH2 group, COH group. Now, at least every amino acids will at least have two pKa values. All right, pKa is acid dissociation constant of acids. Okay. Now, so this suppose this is having pKa1, this is having pKa2. So at least because glycine will have two, and otherwise depending on the type of R group, suppose if it's having like glycine, arginine, all those positively charged amino acids will have like additional NH2 or NH3 group present, or if it's like acidic moieties like aspartate, glutamate, it will have an additional COH group or CO minus group present. So that will again have its own pKa value. Okay. So except glycine, so, so except glycine, all other amino acids will be having an additional pKa value due to its R group or functional group. Okay. So yeah. Now which amino acid will be now these different pKa values and the different types of R group present in an amino acid determines its behavior in a medium. Okay, so its behavior is actually regulated by, to be precise, pH of the medium. Okay. pH of the medium. Now, suppose when pH of the medium, now when the pH of the medium, of any medium, is less than the pKa value, what happens? And when it is greater than the pKa value, what happens? Now, pK is basically the tendency of any molecular atom to donate its proton or electron, or like donate electron or accept an electron. All right. Now, when pH is low, what does it mean? It means OH minus concentration. In OH minus concentration is low, or H plus ion concentration is higher. All right. So this will be reverse in case when pH is greater than when pH is higher. So when H plus ion is at higher concentration, then what will happen here is, for an amino acid, it will be so COH won't try to lose its proton or H plus ion because H plus is already abundant in its medium. But since H plus is abundant, so the NH2 group here, sorry, NH2 group here yeah, yeah. So NH2 group here will be having, it is having a lone pair, so it can actually donate this particular lone pair to an H plus atom ion, forming NH3 plus. Now, but when gradually the pH of the medium increases, or OH minus concentration increases, then what happens? OH minus, because it's higher, it tends to accept H plus ion. Okay. So then the COH group here, present here, can easily donate its H plus ion, thereby forming CO minus group. All right. So depending on the pKa of the groups present over here, the functional group here, the ionizable groups present in an amino acid, the pH of the medium actually regulates the overall charge present in an amino acid. So suppose in this case, let's just randomly take an example that pKa1 is suppose 2 and pKa2 is suppose 10. Now, if the pH of the medium, now considering this example, um, if we are considering this example, okay, just write it. It's pKa1 is 2, pKa2 is 10. Now, if the pH of the medium is suppose 1, what will happen is, in this case, pKa1 is 2. I mean, this is 2. Now, for this particular amino acid, the pH is 1, so it is less than the pKa value, which means H plus ion is already abundant in the medium. So this will remain as COOH. Now, what about this NH2 group? CH, it is less than the value, pH is less than 10 definitely. Now, so what will happen is H plus is abundant in the medium. This will donate its electron to this, forming NH3 plus. So the overall charge on this amino acid will be +1 when pH is 1. Okay. This is just a vague example, random example, just to make the concept clear. Next, suppose now the pH is 6. What will happen? So at pH 6, what happens is pH 6 is greater than the pKa1. All right. So now it has more OH minus, as so this carboxyl group will try to give away its proton. Okay. So this will exist as CO minus, but this 6 is still less than 10 here. So it will remain as NH3 plus. But when we look at the net charge on this particular amino acid, so okay, we are looking, okay, this is R. Okay, R. Now, so this has an overall charge of 0. Okay, net charge of 0. Now suppose what happens when pH is 11. So when pH is 11, then we again have, for 11 scenario, it will again exist as CO minus, will tend to give away its proton. And in this case, again, 10 is like 11 is greater than the pKa2. So that's why this will tend to give away the H plus ion, thereby forming simply NH2 with its lone pair on the nitrogen atom. And the net charge here is -1. In this case, this is 2. Okay. So this is how the now suppose now not R. Suppose this is we are talking about glycine only. Okay. Now if the R group again has an additional charged group or charged moiety, whether it's negatively charged or positively charged, again that will have an additional role to play, which will ultimately affect the overall charge on the amino acid. Okay. So now when we saw that when any amino acid exists in this form, wherein each ionizable group is charged, but then the overall charge in the amino acid is 0 or net charge is 0, overall charge is 0, then this is called Zwitter ion, or the pH at which this amino acid exists as a Zwitter ion, that is the Pi, or is also known as the isoelectric point, which basically refers to, so when uh, in an electric field, this is the pH at which there will be no net charge on an amino acid, and so it will not be moving in an electric field. All right. If it's positively charged, it will be like uh, repel. It will be repelled away from positively charged electrode, and it will tend to move towards the negatively charged one, and vice versa in case of a negatively charged uh, amino acid. But then when it has a neutral, when it is neutrally charged, or it has an overall net charge is 0, then it does not move across an electric field when an electric field is applied to it. Okay. Now this actually plays a very crucial role in determining the property of an behavior of an amino acid in a solution also. Suppose when we want to purify a protein, I think just briefly talking about like chromatographic techniques, when we purify a protein, so the buffer conditions wherein we need to stabilize the protein, or when do we have to do cationic exchange or anionic exchange chromatography and all those steps. So we have to take care that what will be the net charge on our protein of interest, or protein of interest, and that will be determined by its constituent amino acids and the functional groups attached to them. And this is basically determined by the Pi of the protein. Now, if we have to give a net negative charge to the protein, suppose for a net negative charge to the protein and a net positive charge to the protein, what we do is if we increase the pH value, which means OH minus is higher. So as we saw in the previous case, we saw is if OH minus is higher, it will tend to take away the H plus from the carboxylic group, thereby giving it a negative charge. Okay. So if pH is in that way, so net negative charge, net positive charge again is controlled by the pH of the medium or pH of the medium or solution or solvent. We must solve it. So this is what was the briefing before we move on to the titration, characteristic titration curve of an amino acid. So every amino acid, because the R group varies so much in all the amino acids, so every amino acid has characteristic titration curve, wherein these amino acids are actually, so there this y-axis represents the pH range, and this is basically the concentration of OH minus ions. Now here within as the we see that we see different kinds of behaviors. There are inflection points, there's plateau regions, and all these wherein we see these different areas or different behaviors of each amino acid depending on the concentration of or depending on the pH of the solution. So yeah. Now here we see that when at very low pH, H plus is greater in amount. So NH3 net charge, NH2 is existing as NH3 plus, net charge is plus. Now as the pH slowly increases or the concentration of OH minus ions increases, then it this particular COH group tends to lose its proton, giving CO minus group, but then here the net charge is 0. Again, as we go on increasing the pH, then what happens is this is already CO minus, this again loses its H plus, gives away its H plus, forming a net negative charge. Now, so this particular pK1 value is 2.34. pK2 value is 9.60, and from these two values we can actually find the pH at which this particular amino acid, which is glycine in this case, exists as a neutrally charged amino acid. So we can find the pi value from these two values, pK1 plus pK2, take its average, which comes at around 2.34 plus 9.60 by 2, which is 11.94 to 5.97. This is the pi of glycine. Now pi of glycine is 5.97. Now let's just see if what happens when pH is greater than the pi value and pH is less than the pi value. When pH is greater than the pi value, what is happening here is OH minus concentration is greater. And when pH is less than the pi value, just give me a moment. I'm sorry. Yeah. Now, here, yeah, when pH is greater than pi, we have like more amount of H plus ion or less amount of hydroxide ions in the solution. So, yeah. Now, so what we see here, when the pH is less than 5.97. When the pH is less than 5.97 in this area, what we see is there's excess amount of H plus, and it exists predominantly as +1. It has a net charge of +1. And then it has, at higher pH, it has a net charge of -1. Now, so this is what is being shown here, that it is basically existing as at points, at pi values below, at pH value below 5.97. It will be existing predominantly as a positively charged moiety, and at pH values greater than the pi value. So for here, if pi value is greater than the pH, as in 5.97, if the pH is less than 5.97, this will exist as here. See, in this case, it's less than 5.97, the pH value. So it is existing predominantly as a positively charged amino acid. And next is negatively charged amino acid. Okay, if the pH is here, as we see here, when the pH is greater than the pi value, then it's predominantly existing as a negatively charged amino acid. So here, one thing to note also, just a brief thing. When we plot a graph, this is y-axis, this is x-axis. Now, suppose this is A, B, C. So when we talk, this is theta C, theta B, theta A. Now, when we talk about the slope of a graph, which is actually change in the y value or vertical axis upon change in the x value or change in the horizontal axis. Okay, so greater the slope or steeper the slope, greater will be the theta value. So here we will say that theta A is greater than theta B, which is greater than theta C. Okay, considering actually this is also represented as tan theta. So we actually are considering that this is nearly equal to theta, then this will be the case. Now, so here, just steeper the slope, the change in y value would be greater. So this is the significance of this particular thing. Now here in this graph, we see that this area is steeper, very, very steep here. So the slope here would be really, now, so this particular area will be very sensitive to changes in pH. Okay. So this particular region will be very sensitive to pH. Now, here we see that this is a plateau region. This area and this area. This is a plateau region here, wherein it won't be very sensitive to pH. So, but here, even if, so in this particular very steep area, what will happen is, even if we slightly change the pH, it will have a huge difference. As in, if we slightly increase the OH concentration, we see that, see, if suppose at this particular point, at this pH was this, suppose X or Y1. Okay, but immediately after increasing maybe by 0.2 amount, it goes up to this area. Whereas for this part, even after increasing the OH minus by significant amounts, we see that the pH increase in pH is more or less the same. Like there's not a significant increase, but here it's very sensitive to increase in pH. So these areas where the slope is very less, okay, like less steep, very less. So this is actually buffer region, wherein it's very less sensitive, or it can really, even though the OH minus is increased significantly, concentration is increased, it is less sensitive to OH minus concentration. Okay, this area and this area is the buffer regions. All right. Now, this was the case for the simplest amino acid, which is glycine. Now, in case when uh, yeah, here when the R group is much more complicated, or there's any positively charged or negatively charged group here, in case of suppose glutamate, wherein there's an additional carboxyl group present over here. So here what happens is when the pH is too low, so both these carboxyl group have H attached with it, like it hasn't yet donated its H plus, and NH2 will be present as NH3 plus. So net charge is +1 here. Now in case of where it slowly increases, when the pH slowly increases, then which means the OH minus concentration is getting increased slowly, then slowly one of the carboxyl group, this one, especially this one attached to directly to the alpha carbon, being more electronegative, it will have a greater tendency to lose its proton. So it will readily give away its H plus and thereby forming a neutral charged species. Again, when OH minus is increased, then this particular carboxyl group will donate its proton, and with further increase in OH minus, this particular NH3 plus will give away its H plus, thereby giving a net charge of -2 to the amino acid. Okay. Now, so here, so this will have like different more number of inflection points. Also, here in this case, um, yeah. So suppose in case of glycine, we saw that it was simply how were we calculating pK1 plus pK2 by 2 for glycine. Now, in this case, what we will be looking at is the those pKa values, which are basically uh, giving rise to, with a single change in proton, it will give rise to zwitterionic form. So here, in order to calculate the isoelectric point of glutamate, it will be pK1 plus pKr divided by 2. Yeah. So similarly, in case of histidine, again, it's the same case with gradual increase in the OH minus concentration. We have different charges on the overall amino acid. In this case, it will be like it will have the maximum charge of +2, and with increase in OH minus concentration, it will tend to take away the proton, and ultimately at the highest point, it will be existing as a minus, like it will have a net charge of -1 on it. But when we want to really calculate the PI value, it will be basically this average of pKr plus pK2 divided by 2. Okay, in case of histidine. So again, this varies depending on the type of R group or type of functional group present over here. Now, Now, see, these are the whatever we saw earlier, they're the naturally occurring amino acids, and which are actually incorporated into during protein synthesis. These are actually coded by the genetic code, and those amino acids are incorporated into the protein chain. Those are actually incorporated by the ribosomes. All right. But then there are some naturally, like, as in naturally occurring amino, as in which are found in living systems, but then these are actually the derivatives. These are actually derived from those, from the 20 naturally occurring amino acid. Okay. Uh, so here, suppose let's look at the examples, and it does serve specific different kinds of functions in our body, or not only in our body, in general in living systems. So suppose here we have hydroxyproline, which has been modified, which is a modified form of proline here, uh, which is basically which has been found to be present in plant cell walls, and it is also found in collagen connective tissue matrix. Okay. Hmm. Connective tissue matrix. Then we have hydroxylysine, like, which is again a modified form of we can also write here derived or modification, different kinds of modifications which are present on these amino acids. Okay. So 5-hydroxylysine is again a modified form of lysine, which is again present, which has been found to be present in collagen. Okay. Next, we have yes, another modified lysine, which has been found to be present in uh, contractile proteins like myosin. Hmm. Next, we have gamma carboxyglutamate, which is found in prothrombin. Prothrombin is a blood clotting factor. Okay. It is also found in amino acids like as in proteins, especially proteins which bind to Ca2 plus binding proteins. Okay. Then we have desmosine. There are like one, two, three, four lysine residues, which has been modified, and this desmosine is found in fibrous proteins like elastin. Then there's selenocysteine. So in cysteine, the structure of cysteine was this. It was having CH2, sorry, CH2 sulfur attached to H. Okay. Now in place of sulfur, this was cysteine. And in place of sulfur, this has been replaced by selenium. Okay. In this case, thereby forming selenocysteine. So these are some of the uncommon amino acids which definitely serve different kinds of functions in living systems. Okay. Now, next, we looked at the different kinds of monomer units. Now, let's just we will be focusing on how these monomeric units are joined to form, um, you know, dimer, trimer, oligomer. polymer like that. So in this case, when two amino acids are joined together, they are joined together via peptide bond. And so that is called a dipeptide again. So multiple amino acids joined together is known as polypeptide, which again fold in itself, having specific structures due to different kinds of interactions between them, or different exist due to the different kinds of forces acting between the different molecular constituents, thereby forming proper structure, 3D structure of a protein having definite function. Not only 3D, like in general, it gives a structure to the protein which is able to perform a specific function. Okay. So we will be focusing on structure, like very elaborately, we'll be focusing on structure and function. Definitely. In case of function, we will be mainly looking at enzymes and its functions or enzyme kinetics. But then first, as of now, we'll be looking at different properties of peptide bond, how it's formed, and what are the characteristics of a peptide bond. So a peptide bond, now suppose there's one amino acid having R1 functional group. There's another amino acid having R2 functional group. So what will happen here is, let's just write it this way. Yes. Okay. Now what will happen is, this will go away as H2O. This is a condensation reaction. Condensation will occur to give rise to CH. This is the one amino acid. Water is like OH group is gone from here. Then we have N here, which is having an H attached to it. Okay. So this particular bond here is known as peptide bond. So this is basically a dipeptide with only two amino acids present. But there might be multiple amino acids. Now, because this is the representation, this is the all the amino acids occurring here are L isomers or L stereoisomers. So what happens is, there's a this is how proteins are synthesized. So a dipeptide, or suppose that there's a polypeptide. All right. R1 CONH CHR2 again CONH CR3, okay, and so on. So like n times, suppose. Now, but it will go on till suppose like R n plus 1, suppose, and that is again having energy. So because these are all L forms or L stereoisomers, this is how the proteins are synthesized in a cell or synthesized by the ribosomes, wherein it is actually read in that manner only, and this is how the whole peptide chain comes out. So this area where the NH2 is present, this is known as the N terminal of this polypeptide or protein. N terminal, this is a polypeptide. When it takes a specific structure, it will form a protein, polypeptide or protein, suppose. So this is the N terminal, and the very end where only this carboxyl group is present, this is called the C terminal of protein. All right. This is the C terminal. This is the N terminal. for protein. Now, um, so yeah, these are the few things to be kept in mind. Now, so again, so X-ray diffraction studies by Linus Pauling and Robert Corey, they found that the general CN bond, okay, this is a single bond or sigma bond, single bond or sigma bond. Then you have C double N, double bonded to N, which is pi bond. Hmm? Now, what they found, the X-ray diffraction studies, the peptide bond. In a peptide bond, this particular bond is having, like, it's greater, like it's shorter than a sigma bond or single bond length of a sigma bond, but it's greater than the bond length of a double bonded uh, like atoms. Okay, bond length between two double bonded atoms. So, which implies that they do have a partial double bond character. What is happening here is, let's just draw here maybe. So suppose this is C alpha, this is O, this is NH, this is again C alpha for another amino acid. Okay. Let's suppose this has a lone pair. Happens is it goes in there, then again when it comes here, it again comes there, then this goes back here. So this is how it takes place. So what happens here is minus plus, like that. So ultimately what we see is this kind of partial double bond character wherein there's a partial negative delta minus charge on oxygen atom and delta plus charge on nitrogen atom. Okay. Wherein this electron is delocalized, kind of, there's a partial double bond character in this region. So this also suggested strongly that these four atoms, O, C, N, and H, are reside in a single plane. Okay. Reside in a single plane. So these are all coplanar. Okay. Now, these X-ray data indicated a resonance between the carbonyl oxygen and amide nitrogen. Carbonyl oxygen, this one, and amide nitrogen, this one. Okay. So these are all resonance intermediates. Okay. This is one intermediate and another. So these are the resonance structures giving a bond length that exists between a sigma bond and a pi bond. Okay. So yeah, consequently, the peptide bonds, that is what is written here. Over here, that the peptide bond length is only 1.33 angstrom, which is shorter than the usual CN bond length of 1.45 angstrom. Okay, this is the sigma bond we are talking about, as in the C alpha N bond. It is, however, longer than the value of 1.25 angstrom for the average CN double bond. So it was found that the peptide bond appears to have nearly 40% double bonded character. And as a result, rotation of this bond is restricted and the 6 atoms, okay. So here when we talk about these ones, now this is a sigma bond, this is a sigma bond, all right. Now, so they are able to freely rotate, the atomic moieties here attached here are freely able to rotate around this bond, all right. But in across this, because of having a partial double bond character, the movement or rotation around this particular bond is restricted, somewhat restricted. So this is what is written over here. So because it's like restricted, so it is supposed to be lying in a same plane. Suppose if there's a flexibility of or rotation is possible, it's flexible, then there's a tendency of the different kinds of functional groups to be present out of the plane or going into the plane like that. But then because the double across this partial double bond character, there's a restricted movement of moiety. So the atoms which are actually participating in the uh, peptide bond formation, those are those ideally should be coplanar or lying in the same plane, which is what has been observed also. Yeah. Now when we talk about, now this is a peptide bond, this one. Now this is one now this part belongs to one amino acid. Okay, suppose amino acid 2 and this part belongs to another amino acid. So when the C alpha or whatever group is attached there, uh, they lie in an opposite configuration, then it's a trans form, a peptide bond, and otherwise it's a cis form. Okay, and both the C alpha are positioned in this way. So it has been found that trans form is intrinsically favored energetically. Now this must be definitely like probably because lesser, see here both the, because this isn't only C alpha, it has like H and other R groups attached to it. All right, suppose this is one amino acid, this is another amino acid. Now there will be repulsion or steric hindrance or steric effects between these two moieties or the functional groups of these attached with the alpha carbon of these two amino acids. But then if they are placed away or opposite to each other, then there will be lesser steric hindrance, okay, or lesser steric interactions. So this has been found that the trans form is intrinsically favored energetically. Now, if we talk about in case of proline, what happens is proline has a cyclic chain. Okay. Uh, so in case of proline, if after a peptide bond, so this is a peptide bond here, and then after that, if suppose this is this amino acid 2 is actually a proline, then what happens is whether it's cis or trans, the stability or intrinsic stability is comparable because it's a cyclic five-membered ring. It doesn't have freely available functional groups attached to it. Okay, so this is what is written here. Cyclic side chains diminishes the interatomic repulsion, which can be seen in these freely attached functional groups like R1, R2 in these two different amino acids. All right. So yeah, this was this portion for today's class. Next, again, we will be we'll be talking about uh, different kinds of rotation or dihedral angles, as well as we will move on to different structures of a protein, how the amino acid uh, type of kind of amino acid and the interactions between its functional group or interactions between different amino acids leads to the formation of different kinds, like very different kinds of structures in proteins. So that's all for this part. Stop sharing. So yeah, bye. Thank you.